Right, Dr. Paddison, are you ready?
Yeah, thank you for that kind introduction, and I'd just like to thank the organizers for inviting me back this year, despite the fact that I was on the same plane with Cameron, who mentioned yesterday that we had engine problems and actually had to turn around, and so we couldn't make it last year. But I would just like to say it's been a terrific conference so far. I've certainly learned a lot, and I've gotten a taste of Hong Kong. Literally, we had a great dinner last night, and I had a lot of fun. So I certainly, this is on my list of places to come back to visit. So my lab focuses on functional genomics approaches in a variety of systems. And what we like to say is that we put the fun in functional genomics. It's not always fun, as many of you may have found out the hard way, but we apply functional genomics to human and stem progenitor cells, and there's really three moving parts to my lab. One involves sort of the maintenance and generation of functional genomic libraries, and then we have biological arms. One is focused on developmental biology, where we look at lineage transitions and cell identity pathways, and the other one that I'll focus on today is a cancer focus, where we're looking at new therapeutic targets, new cancer biology relevant to vulnerabilities induced by oncogenic stress. So the main screening paradigm that we use for our functional genetic studies was developed a number of years ago by me and many other people back in the days when I was in grad school. And it's a pooled screening approach where we actually grow oligos on chips, we cleave them off and create these pooled libraries, and then in turn create pooled viruses that can be in turn used as screening pools in target cells. And nowadays, the way we read out sort of screen phenotypes is through deep sequencing and deconvolution of the library after we've performed our phenotypic assay. The simplest one simply being outgrowth, where we examine the distribution of, for example, hairpin sequences or sgRNA sequences in a population at day 0, shortly after infection, and then after an extended time point after outgrowth, then we look for gross changes in representation of the library. The fun part nowadays, as opposed to back in grad school, is that this oligosynthesis part has actually gotten a lot better and more freely available. So now we're able to actually do iterative screens where we start with a genome scale library, do one iteration, and then we can go back and do smaller iterations through oligosynthesis. And we have all the protocols for doing this if anyone is interested in building custom sets. So today, I'd like to give you an overview of our target discovery program that actually dovetails quite nicely with Stephanie's talk, because actually one of the targets I'll mention is the very target of the drug she was just talking about, WEE1, that actually popped up in a funny way from one of our screens. And those screens were gene editing screens. And so I'll talk about some of the highlights, but also some of the challenges we face doing CRISPR-Cas9 screens. So our focus is glioblastoma. It's the most common and lethal brain tumor in adults. There are about 12,000 cases of glioblastoma in the US every year. It's a very lethal disease. None of the current therapeutic modalities work very well: surgery, alkylating agent, radiation. And so there's clearly a desperate need for new therapeutic targets. Like other cancers, there are well-known signaling pathways that are altered in this cancer that you really see in most of the tumors, including the RTK RAS, PIK3CA pathway, P53, the RB axis, and also IDH1 mutation, which tends to occur in lower grade glioma. And so when you think of precision medicine for this cancer, with the current state of clinical practice, it might look something like this, where you have a diagnosis, and then you have surgical resectioning followed by tumor profiling. And then you might see molecular features that we all know are associated with the disease, and then you hope that that would allow you to inform what you do after or during treatment with a standard of care. I think one of the questions that I have in general, being a screener, is what sort of targets are we really looking for, and whether our current notions of precision medicine are actually adequate for coming up with better therapeutics. And certainly approaches like Stephanie's are really cool and may actually dramatically change the profile of certain cancers with existing therapies. But I think one of the things that is true is that we've been spoiled a little bit by our approach to precision medicine from the use, for example, of things like Gleevec, where you have a novel oncogene target that was very susceptible to the drug and resulted in a very dramatic effect, which hasn't then really translated to solid tumors. And so one way of looking at this approach, where you're trying to target oncogenic drivers and identify them in patient samples, is sort of shooting the drivers. If cancer is an out-of-control stagecoach, you're trying to shoot the oncogenic drivers. But unfortunately, for glioblastoma, like other solid tumors, this approach has really failed to produce a survival benefit. And there are many reasons why this could be. I'm not going to argue that it was necessarily a failure of the drugs themselves. Obviously, there are issues with clinical study design and that sort of thing, but it's still formally possible that these are simply not the best targets, and maybe we should find different targets. And so that's what we do. And so this is our general screening paradigm for this cancer. We actually pluck cells that are stem-like cells, glioblastoma stem-like cells, from patient isolates, and we grow them in defined conditions in vitro. And then we do compare and contrast studies with not necessarily a cell of origin, but certainly a tissue of origin control and a sort of a non-transformed control in neural progenitors. And so we basically perform screens. We look for hits, in one case, sort of just generally lethal genes, and we do comparisons. We try to integrate this information into networks derived from patient databases. And then we try to identify the things that, in this case, tend to kill the GBM cells, but spare the normal cells, with the hope that we can create sort of a in vitro model for therapeutic window. The fun part is that these same models that are derived from patients can allow us to do mechanistic studies and sort of pick apart the biology underneath or underlying the lethality. And so we have a bunch of papers where we've picked apart different targets, and we like to sort of just be sure that the biology is real, and these systems have allowed us to do that. Now, driving this are these glioblastoma stem-like cells, and they're great because we can derive them in serum-free, defined conditions on laminin-coated plates, and they retain karyotype and gene expression profiles of the original tumor and can regenerate the tumors in the rodent brain, immunocompromised rodents. But I think the thing that illustrates why they're sort of useful and cool is this, which is a principal component analysis of gene expression profiles of these isolates from glioblastoma that are grown in defined conditions, shown here, that sort of cluster together, or in serum, which is what, as you know, what most of us grow cell lines in. And as you grow these guys and isolate them in serum, they actually start segregating away from the original patient tumor and start segregating towards the garden variety glioblastoma cell lines that are more sort of similar to HeLa cells. Forgot that slide. But you can also see this by karyotype. And so the karyotype of these guys is very similar to a HeLa cell, which is crazy, and these very much resemble the original patient isolate. That's not to say these things are perfect. So if you look at the original patient tumor, this is a simple resection study where they're taking these sections out of the tumor and then doing a simple mutation profile and trying to look at clonal analysis and sort of infer what a founding clone a mutation spectra might look like. You can see that there are a number of subclones, and when we isolate these cells from patient tumors, what I think we're actually doing is isolating a subclone. So we're not retaining the heterogeneity of the original tumor, but in some ways that's not bad because it simplifies our life experimentally. So these are not perfect models, but I like to think that they're useful. And one of the reasons I think they're useful is that we can derive a lot of them and have a bank of them and actually have them retain these molecular features of the original patient tumor, shown here as the classic 4 subtype classification of GBM, and we can find the alterations and gene expression networks that are here within these isolates. And we can even, in some cases, match pre and post therapy samples using warm autopsy programs. So we collaborate with a lot of people around the world to create these banks of cells, and over the years we've done a lot of screens to come up with therapeutic targets in these cells. And I won't go into all of this work, but I'll just highlight a little bit of it. So some of the work we've already published has shown that these cells have vulnerabilities in kinetochore microtubule interactions where they exhibit lethal kinetochore microtubule instability. Basically, the end-on attachments of chromosomes and microtubules are unstable, and this is driven by RAS or RTK-RAS activity. They have chromosome alignment defects. They have defects in centrosome clustering, and even 3' splice site recognition, which is, we found, driven by high MYC activity. So we're able to start to propose or nominate potential therapeutic targets. And this is an example of the one that's driven by MYC activity, shown in vivo, where we're using these MYC high isolates to, in combination with an inducible hairpin, to create an orthotopic tumor that we can then inhibit by inducing knockdown of this gene. You can see the tumor dramatically goes away, and there's dramatic survival benefit, at least at this time point here. So overall, what we're trying to do in my lab is really the first part of what's shown here, where we have this brain tumor in vitro resource. We do the functional genomic screening. We've tried to validate these targets mechanistically, and then we try to come up with a rationale for why this could be a good GBM therapeutic and try to associate it with an oncogenic activity. We also then work with Jim Olson's lab at the Fred Hutch, who's a neuro-oncologist and pharmacologist, to try to evaluate candidate targets and then try to get them into some sort of therapeutic development pipeline. which, as I'm sure many of you know, can be very challenging for academics. And so we're constantly looking for academic collaborators and pharma partners to do this. In addition to that, I think, one of the issues that comes up whenever you do cancer target screens is that there are a lot out there. There are a lot of screens, there are a lot of claims about what's a cancer target. And so one of the things we're doing now is actually creating mouse models for the targets that we're proposing to actually confirm therapeutic window more broadly across many mouse tissues, and also ask the question of whether we can translate the target to other cancers, which I think will likely be possible. So one of the things that we've been doing in the last few years is trying to simply ask whether, so most of the work that we've done in the past has been with RNAi libraries, which is sort of my background. But with the new kid on the block, we were interested to know whether CRISPR-Cas9 was going to work in a similar way, in a different way, in a better way than our previous work with RNAi. So as you guys know, CRISPR-Cas9 has been quite the new kid on the block in the last few years. And the way we've been using it is using the nuclease competent version of Cas9 to actually create site-specific alterations in the genome, inducing double-strand breaks, and then allowing the cells to do sloppy repair using non-homologous end joining, which then creates an indel, insertion-deletion mutation, which in turn will create some of the time a frame shift and a premature stop codon, thus inhibiting the target gene. So we were interested whether this would work at a reasonable frequency in our primary cells, and we tested a number of vectors, and we ended up going with this all-in-one Cas9 vector that also expresses the sgRNA as a lentivirus. And whenever we use a new vector or use a new library, we basically take it through sort of the paces, sort of, how does it work? What is the sort of the response window? And in this case, we were simply targeting EGFP, a single integrated copy of EGFP in neural progenitors, our control cells, and also the GBM stem-like cells. And you can see here that the response is very dramatic. So these are the control cells that have high GFP, and then these are the ones in red that have the sgRNA. There's almost complete ablation of expression of GFP, but one thing we noticed right away is that it actually took time in our primary cells for this to work. It wasn't instantaneous, and it seemed to be somewhat of a heterogeneous effect that would sort of accumulate over time and then, boom, present you with a big effect. So it's a little different than using RNAi. We're interested to know whether we could target endogenous genes. This shows targeting p53 in neural progenitors, where we're inducing or we're stabilizing p53 with doxorubicin, and this shows profound loss of p53 expression after sgRNA treatment. And we were most interested, of course, to know if we could target an essential gene. And this shows the essential replication gene, MCM2, targeted with 3 different sgRNAs at these exon sites. And we can show in both our neural progenitors and GBM cells that we see significant loss of cell growth 13 days post-selection after viral infection. One of the, getting back to this notion of frame shifting, one of the questions that really comes up with phenotypes produced by nuclease version of Cas9 is how penetrant the effect would be. These pie charts show sequence results from indel formation at particular target sites. And so each slice is a frame. And so if you would expect indels to be repaired in an unbiased way, it would essentially look like this one. And so a third of the time you'd end up with a very small insertion or deletion of a few bases, and perhaps a near wild type protein activity. But that doesn't seem to be the case. The white is the wild type frame, and we see frame shift bias. These are all things that scored in our screen, and we retested them by deep sequencing here. And so it's unclear right now whether positives arising from our screens are actually resulting partly from frame shift bias at the target sites. That'll be an interesting question to resolve in the future. So where we currently are with our screens is here. So we, a few years ago, published an initial stab at doing genome-wide screens in GBM stem-like cells and neural progenitors, and we've since progressed. So we now have done full genome-wide screens in 3 isolates that have been genomically profiled in 2 neural progenitor cells targeting about 18,000 genes. And that's what this looks like. So this is the equivalent of 4,000 10-centimeter plates. And that was a lot of work. And so that's one bay of my lab. And that was mainly done by one postdoc who's pretty amazing. But it also speaks to the utility of our cells, that we can actually grow them in these mass quantities to do these sorts of large screening efforts. This is what the data looks like. So in this, again, in this case, we're comparing the representation of the library at a day 0, and in this case, day 21 or day 23. And then we simply computationally count the reads of each population and compare them. And so in this case, the things in red are reads that are going up as the cells grow out, and the green ones are the ones that are going down, and you can see MCM2 is among the green. And so these screens actually work quite well in this format. And our ultimate goal, again, is to basically compare these 2 green populations, what's required here, but not here. And so when you look at those green populations, you see the usual suspects. We see, when we do gene set enrichment, we see things like translation, RNA splicing, et cetera, again suggesting that these libraries work very well and efficiently for identifying essential genes. We also see things in our case that are relevant to neurodevelopment that might be sort of tissue specific, which is nice. And so we, a new grad student in lab has just picked up this project, and what we've now done that we hadn't done before is comprehensively retested all possible GBM lethal genes, even the ones that scored weakly. We created this big category of about 1,000 or 900 genes, and then we did the retest in all of the original screen isolates. And this is an example of how we then tried to define the ones that are more specific to the GBM cells. This just shows you the sgRNA-seq for the GBM and the neural progenitors, and the ones in black are the ones that tend to be more negative in the GBM cells. And so Pia has now then made a secondary screening pool of 58 high priority targets, some of which are shown here, and then done this tertiary screen in about 20 brain tumor isolates that have been genomically profiled. So the concept is, we can't simply do these genome-wide screens in all of our isolates because it's just too expensive, but what we can do is make refinements and then test hypothesis of maybe the hits that are specific to this cell type or this isolate are actually associated with the genomic alterations that are contained within those isolates. And we can start to answer that question by doing these smaller screens in a bunch of isolates. And that's what some of this data looks like. So we've been able to do these smaller scale screens and actually start to identify candidate synthetic lethal genes because we know the molecular genotype of the isolates that we're screening, and so we can come up with possible predictions. And one example of that is here. And so in the case of this guy, PKMYT1, it's a WEE1 kinase. It's we found that it's generally lethal in our GBM stem-like cells, but then over here, this gene FBX042, an E3 ubiquitin ligase, seems to segregate with activating mutations in PIK3CA. So that could be pretty cool, and we're currently pursuing that. But here's sort of a partial list of our inferred synthetic lethal predictions. And so we have some that we think are actually caused by overactivation of both the RTK and PIK3CA or PI3 kinase pathway, EGFR amplification, MYC activity, or PIK3CA mutation, or in combination with PTEN. So these are some pretty interesting genes. Some of them do have druggable activities. We're currently retesting them now. The other interesting thing that we can start to think about is what I like to think about, sort of the dashboard of synthetic lethality in these cells and in this cancer. So the question starts to become, what can be synthetic lethal with what, at least what we can see. And what we can actually start to do is cross things off the list based on what we know is altered in the cells that we're retesting the small pool in. And one of the interesting things, or two of the interesting things, are that we don't find synthetic lethals with the p53 pathway or the RB axis. And so our prediction moving forward is that the synthetic lethals that we tend to find are going to be up here with MYC, RTK RAS, PI3 kinase pathway, but there are certainly things we're blind to, and so none of our isolates actually have IDH1 mutations, for example, because those cells don't grow well in vitro. And so we're also using Cas9 to help test those predictions. So one of the way we're doing it is actually engineering our neural progenitors to put the oncogenic drivers and tumor suppressors into those cells via Cas9, but then we're actually using the RNP version of Cas9, the in vitro complex, to generate indels in the candidate synthetic lethal genes at high frequency. And in this case, we're doing a clonality test where we can actually sequence clones from these tests where we're manipulating these defined transformed or partially transformed cells, and then ask the frequency at which we see wild type, heterozygous, or biallelic targeted genes. And this is one of the actual problems with doing these CRISPR-Cas9 screens, is that when we do them, we actually don't know, for example, if we're looking at a lethal gene, whether they're het lethal or homozygous lethal. And moreover, if you're dealing with a line that has a crazy karyotype, you might have a target that has 6 copies or more. And so, that working that out can be a little problematic. So I'll briefly mention PKMYT1. And so in our first publication, what we were actually able to show is, it's also called MYT1. MYT1 is a WEE1-like kinase that acts redundantly with WEE1 to add inhibitory phosphorylations onto CDK1 cyclin B to block its activity and help coordinate entry into mitosis. And so previous work in mammals in particular has not appreciated this redundancy between these guys. Work in HeLa cells, for example, has mainly shown that WEE1 controls entry into mitosis, and inhibiting MYT1 does very little. So MYT1 has sort of been ignored from this. But to make a somewhat long story short, what we were able to show is that MIT1 and WEE1 act redundantly to phosphorylate tyrosine 15 in particular on CDK1 as cells are entering, starting to get close to mitosis to help facilitate it. And so when we knock out MIT1 or WEE1, for example, in the cancer cells, they end up piling up in mitosis. And we think the reason is that when we transform our neural progenitors or other cells with things affecting EGFR or PI3 kinase pathway, we actually see a reduction in phosphorylation. So the baseline of these phosphorylation events goes down. We don't know why exactly, but we think it causes a breakdown in redundancy between these guys that causes the vulnerability. And that data is actually shown here, where we are using the same WEE1 inhibitor that Stephanie was just referring to, and we're examining the transit time through mitosis. And in our neural progenitor cells, we see that knocking out MIT1 actually confers very little change in transit time, and similarly with WEE1 inhibitor, but together they dramatically pile up in mitosis. But then in the cancer cells, we actually see that knocking down or out either one actually confers sensitivity, and we can see that in looking at cytokinesis failure or cell death during mitosis. And in vivo, we also see a somewhat dramatic effect, although the survival curve is somewhat modest in its change. You see this dramatic phenotypic effect with this most aggressive, basically the most aggressive patient isolate we've come up with, which is shown here, where it just results in these very infiltrative tumors. And when we knock out MIT1, we see these more defined tumors where we do get an extension. And right now we're trying to, of survival, but right now what we're trying to actually ask is whether this is a real, whether these are residual cells that have wild type MIT1. One of the issues with any of these technologies is that the cancers find ways of defeating them, or whether these are truly deleted for MIT1 and it has changed sort of the profile of the cancer. It's the new version of PowerPoint. So in general, just sort of to sum up what we're doing with these things and sort of our view of precision medicine is that the way I view it is that we're trying to change the flow of information. This isn't to say that going from patient isolates and sequencing them and coming up with new, more sophisticated ways of analyzing the data won't work, but I think we're still blind to many of the things that we're finding that you simply can't predict from the sequence analysis alone. So, for example, almost none of the targets that we find are drivers. And so it seems like our system is teaching away from targeting drivers. Now, of course, we're not using drugs. And so there's a fundamental difference between what we do with targeting genes in the genome versus targeting drugs with small molecules, as you guys know. So there are big differences, but nonetheless, we are finding targets that seem to be downstream of the activity of these oncogenes. And our hope is to actually come up with patient classifiers and new therapies that will then inform the sort of molecular profiles that we find in the patient tumors. And so, of course, it's more complicated than just that. We still haven't dealt with tumor heterogeneity. There's also the combination sort of therapies that Stephanie was referring to, and there's also, in this case, delivery to the tumor. So these are big problems, but we are working with collaborators on those. So with that, I'd just like to say thank you for your attention, and thank you to the members of my lab and all of our collaborators for the wonderful work that they do. Thank you.